When you were younger, I bet you were very curious. In fact, I know everyone started out their life very curious. You were curious before you even remember. You were curious about your hands, about your feet, about every single thing on the floor and what it tasted like. And I know this because I have four children that I raised, and now I have two grandchildren that I get to see once in a while. And when they're at my house, they definitely love to put stuff in their mouth. So I kind of know that. So biologists are also very curious. And because of their curiosity, they like to learn more and more about life, since biology is the study of life. And so biologists use a process known as the scientific method to study living things. So the scientific method is a series of steps that allows us to learn a little bit more about living things and about the living things that we care about. So it starts out with some observations that allow us to make specific hypotheses. A hypothesis is an explanation for what you're seeing or for what's occurring. You then make a prediction and you experiment on whatever you're looking at. So you run an experiment. That experiment gives you data points. The data points you analyze and you determine if your hypothesis has been supported or not. Sometimes your hypothesis is supported and that's great, but sometimes it's rejected. That's not bad. That means you learned something and maybe you'll come up with another hypothesis that you can then run another experiment on. It can become very tedious when you're working and working and working and not getting the results you want, but that's the wonderful thing about science and the scientific method. We're always making sure that whatever results we have are correct results. So like I said, scientists are curious, and so they are constantly trying to figure out, and by scientists and I'm a scientist as well. So we are trying to figure out what is going on with living things. And we use our own senses, our knowledge, as well as instruments to study living things. We use inductive reasoning to come up with a hypothesis. Inductive reasoning occurs when you use your own creative thinking along with facts. So it's not just some willy-nilly creativity. We have to have facts to make it a complete hypothesis. A hypothesis has to be able to be falsifiable and it has to be able to be testable. So an example of inductive reasoning. I studied cockroaches. How fun is that, right? I am a Western Illinois University graduate, and I am the only one at the time that ever studied cockroaches and their pinworm parasites. So I was the first, and there are students behind me that have jumped onto my research bandwagon. So when you go to university for your master's, a lot of times you can jump into a research project that is currently going on, and you can come up with your own study within that research. I, on the other hand, decided to start the process. I wanted to study cockroaches. I did not want to study cockroaches and their pinworms. I actually wanted to study the immune system, but things change. And my advisor suggested I grab some cockroaches, cut them up, and see what was inside. And so I went out to the greenhouse, I grabbed some cockroaches, and I killed them. I killed them very humanely. I used either a kill jar or I froze them. Cockroaches are very small, they freeze very quickly, so they did not suffer. And then I cut them open and I looked in their gut or in their digestive tract, and I found pinworms. And he said, well, I'll work with you. And so me, Dr. Meager, and Dr. McCravey started working on this project. And I did a lot of pre-work. And then after doing so much work, learning how to sex a cockroach, learning how to figure out an age for a cockroach, and seeing all the worms inside the cockroaches, I finally used inductive reasoning to come up with two hypotheses. The first one was sex influences pinworm infection levels in Australian cockroaches. That's what you're seeing on the side over here. The second was age influences pinworm infection levels in Australian cockroaches. And I will not give away what the results are yet. So once I had my hypothesis, I then used deductive reasoning so that I could test my hypothesis. So inductive reasoning uses the if-then logic. So an example, a scientist might reason if organisms are composed of cells, then examination of an organism should reveal cells. Makes sense, right? So my deductive reasoning. If sex influences pinworm infection levels in Australian cockroaches, then males and females will have significantly different levels of pinworm infections. Makes sense, right? So if age influences pinworm infection levels in Australian cockroaches, then, and I'll let you think about this and come back to me and tell me the last part of that deductive reasoning, okay? So you're on a cliffhanger now. So once we had our hypothesis and we had our if-then statement, we were able to then run a test. So there are different types of tests that you can run. But no matter what you do, you have to have good experimental design. Experimental design is how you have designed that experiment. And so for me, I used bottles. So just regular soda or water bottles. So this is what a bottle looks like, right? So there's the bottle. This is the neck, and there's where you drink out of, okay? What I did was I cut this bottle so that there was a bottom half and a top half, right? I then, here's the bottom half. It's a little wider now for some reason, but whatever. And I stuck the top half inside of it. So this neck now was basically a hole where cockroaches would fall into the floor. I put Vaseline on the walls. So this stuck in there. There's Vaseline on the walls. Cockroaches don't like Vaseline. And then down here, I put baked sugar water. The first month, I caught 125 cockroaches. And that was, I baited the traps, I put them out. I had 125 cockroaches. And that was in November, okay? Came back, and in January, I did the same thing. And I caught one cockroach. And my first thought was, did I kill all the cockroaches? What? Like, that's crazy because I'm pretty sure cockroaches breed really fast, right? So I was really confused. I didn't know what happened. Went out again, set traps, and so this is still January. Caught 0 cockroaches. I thought, oh my goodness, what did I do wrong? We went back to the drawing board. And so I got this idea of how to capture cockroaches by a professor in the fisheries department, Dr. Spear. I asked him what he did because he told me, set everything up like this, but he didn't tell me how much sugar or water solution to use. But apparently my sugar water solution was thicker than his, and apparently cockroaches prefer thinner sugar water solution. Who knew that they were finicky, right? So I spent the next 3 months, February through April, testing sugar water solutions. And every month, so I'd set the traps out 5 days a week in the morning, and the next day I would come and get them. I would count the number of cockroaches, I would let them go, and then I would set them back out. So I did this every day, 5 days a week, and there was a statistical significant difference between my traps and Dr. Spear's traps. It wasn't about the bottle shape didn't matter. What did matter was his sugar water solution. They preferred his. So I again restarted my research in June with his solution of sugar water. And then I studied the cockroaches for 1 full year, 2 months out of every single quarter of the year. So experimental design is no joke. If you mess up that, then you're going to have to start over with everything, which is not fun. A lot of times in experiments, we have 2 different groups depending on what we're studying. So in my research, I wasn't studying the effects of something. I just wanted to know what type of worms, and if cockroaches that were male had more, or if cockroaches that were older had more, stuff like that. So I didn't have a control group. But if you're studying chemicals or anything like that on an organism, then you're going to have a control group. A control group is a group of organisms that receives no treatment. And then the experimental group is going to receive whatever that treatment is. And so we often use models instead of the actual subjects. This is very important because you can't study, you can't use a chemical on humans. We can't study, or we can't test on humans in general. And therefore, we have to have some type of organism that we can test. And then sometimes when it's something more about the environment, we can use computers to study, to model the system. So when we run an experiment, we're going to get data. We're going to receive data points. My data points in my research were the number of pinworms, the type of pinworm. There were 2 different species. The size of the back. I believe it was left femur. It might have been the right femur, but I think it was the left because I'm left-handed, and so I always do the left. The sex, and if they were adult. Oh, hello. What happened there? If they were adult or juvenile. Those were my data points that I chose, or that I had to write down for every single cockroach that I studied. Data points So I would put that information in, and then I could run statistical analyses on those data points to help me determine if size or sex was influencing pinworm infection levels. Once we were finished, it took us, so I finished my research and I received my master's degree in 2010. But the research kept going, and they actually worked on it for years afterwards. And eventually, when you do research, you like to publish. And so we published in the Journal of Parasitology in 2019, so just this last year. Dr. Meager was my advisor. Dr. McCravey was a second advisor. He received first person because I said I don't have time to do all the extra work right now. So I said, if you want to finish it, I will let you be the first person on the research, and that's why I'm second person. And so my title of my undergrad and grad research were a little bit more cute. Size matters, because that's what we found. It wasn't sex, it was size that matters. And so larger cockroaches had more worms. It didn't matter if you were male or female. So kind of cool, right? I'm going to stop here, and I will get into, because we only have a few more minutes, but this has been an 18-minute video. So I'm going to stop here, and then we'll go through an actual experiment together. Okay? Bye.